So I'd like to invite our next speaker, Paul Kotzbauer. So Professor Paul Kotzbauer is a professor of neurology at the Hope Center in WashU in the USA. And he's going to be speaking about the therapeutic approaches to restore PLA2G6 enzyme function in INAD. Thank you, Paul.
Thank you, Manju. Can you see my screen and hear me okay?
Yes, sir. We can see your screen and we hear you well.
All right, thank you. I also would like to thank the organizers for inviting me to present at this meeting, and thank you for putting together such a wonderful meeting. I'm going to today talk to you about therapeutic approaches focused on restoring PLA2G6 enzyme function in INAD. And we began this project several years ago, and I actually presented on some of our data at the last symposium, and this will be kind of an update with kind of a new perspective as well on where we might go with this approach. So I think you saw this diagram before, the PLA2G6 gene in Ahad's presentation. It shows all the different mutations that were identified in the initial genetic studies. There are now even more. And what you see is there are a number of quite a few mutations causing missense amino acid changes throughout the length of the protein. It's an 806 amino acid protein. And I want to talk a little bit about the enzyme function of PLA2G6. We don't understand it completely, but we know it as a phospholipase. And while some phospholipases use very specific species of phospholipids and hydrolyze specific acyl chains to produce specific fatty acid products, our studies of PLA2G6 indicate that this particular enzyme does not have much substrate selectivity, in that it can hydrolyze different classes of phospholipids and different acyl chains as well. And while kind of the classic phospholipase A2 reaction is shown here, where this acyl chain is hydrolyzed, we found that PLA2G6 can also utilize the lysophospholipid product as a substrate to produce another fatty acid, and can also start with a phospholipid and hydrolyze the SN1 acyl chain, in a sense having phospholipase A1 activity as well. And we've utilized that. I mean, these are all studies in vitro, and this is an enzyme reaction that's not easy to study in vivo. But by understanding this, at least in vitro, we can think a little bit more about its function, and also use this to refine our approaches to study therapeutic approaches. And so we've looked in, many years ago, we used radio-labeled phospholipid substrates to measure the activity of the enzyme. And when we look at the wild-type enzyme, we can see easily measurable activity. And if we look at this is producing recombinant protein, where we've introduced mutations that have been found in INAD patients, produce recombinant protein containing those mutations. We find that the vast majority of them are deficient, that they have very poor activity in these assays using radio-labeled phospholipids. We've looked in mice in terms of developing a mouse model and thinking about kind of approaches to develop therapies. And so we've described many years ago a mouse model that's different than the mouse model that Ahad and Manju are using for their gene therapy studies. This mouse model has a targeted mutation that deletes exon 9. It's been shown to be a null mutation, meaning there's little PLA2G6 protein, essentially no PLA2G6 protein that's detectable. These mice do well for many months, and it's only about 13 months when you can see clear deficits in their motor function that we can measure on rotarod and other tests. What's striking though is, and this fits with Manju's description of the clinical features of INAD, is that these mice, when you look by histology at the central and peripheral nervous system, they have the defining feature, defining change of INAD, which are these neuroaxonal spheroids that you can see with ubiquitin, anti-ubiquitin antibodies. And on EM, they have these phospholipid, membrane accumulation. These neuroaxonal spheroids contain accumulating membranes. And what's striking is that you can really see these progressively accumulate throughout the central and peripheral nervous system, affecting, at older ages, almost all areas of the brain and spinal cord as well. When we think about what's happening in the disease, in terms of specific molecular consequences of impaired PLA2G6 enzyme function, we think in some ways, in kind of simple ways, that this is an enzyme that normally converts lipids to fatty acids. And so predicted consequences would be substrate accumulation, so phospholipids and lysophospholipase, which appear to be the primary component of the neuroaxonal spheroids that we see. I have argued for rather than a specific signaling role, for a homeostatic role of PLA2G6, which is also supported by some cell culture experiments. What we can't see, won't see under the microscope, is a deficiency of the PLA2G6 products, and that would include free fatty acids. And without, with local deficits in these free fatty acids, you can have restricted availability for lipid synthesis, remodeling, and even beta oxidation for energy production. Another striking feature that we've observed in terms of the effects of the mutations is that these proteins containing INAD-associated mutations don't function well when we introduce them into primary neuronal culture, so primary neurons, in terms of not being able to reach the normal localization that we see for the wild type or the wild type PLA2G6 protein. And so we've done this in a variety of ways. And if we look at the native or wild type protein and label it either with antibodies or by fusion to a fluorescent protein, what we see is a very compartmented localization, and it's primarily in processes. So if we stain the neuronal processes with an antibody tubulin and co-localize, we see these punctate accumulations of enzyme aligning with the neuronal processes. And what was striking is that we've looked at a variety of mutations, and essentially all of the ones associated with INAD disrupt the normal localization pattern, and that the enzyme is either localized more diffusely, primarily in the cytoplasm, or it has a compartmentalized localization, but restricted to the cytoplasm and not getting out into neuronal processes. We've looked for co-localization with various subcellular markers, and we essentially don't see any specific data that helps understand its subcellular localization. And so a basic model in thinking about disease pathogenesis is that PLA2G6 normally lives out in neuronal processes and axons and dendrites, where it converts phospholipids and free fatty acids. When you have mutations, you lose catalytic activity, and you also lose the ability of the protein to be transported, to be localized in its appropriate home. And without enzyme function, you get phospholipid accumulation in neuroaxonal spheroids. And while this may be primarily a loss of homeostatic function, it's also quite possible that this enzyme plays a role in transport, that there's a huge amount of transport back and forth between the cell body and axonal processes, synaptic terminals. And so the role of the enzyme in these transport processes is also quite possible. So thinking about therapeutic strategies, we've actually pursued, to some extent, all of the ones on this list. You've heard about gene therapy. Enzyme replacement therapy is another approach that we've considered, but have not actively pursued, where you could potentially find ways to engineer the protein in a way that would allow delivery of the protein to cells in the nervous system. And what we consider at this point would need to target the enzyme to the cytoplasm. Approach I'm going to focus on today is to improve the function of mutant PLA2G6 proteins, and I'll talk more about that in the next slide. But other strategies would be, can you think about downstream consequences and come up with ways to stimulate other enzymes to compensate for the loss of PLA2G6 function. And so we've also thought about those approaches as well and pursued some of those. Challenges, of course, in all of these approaches, whether it's significant challenges and the ability to target gene therapy throughout the brain, similar challenges for enzyme replacement therapy, which is probably even less well developed in terms of targeting at least proteins or enzymes that are not lysosomal enzymes. And we'll talk about the challenges of the pharmacologic chaperone approach as well. So our pharmacologic chaperone approach seeks to, the rationale for the approach is that many of the mutations change only one of the 806 amino acids in the protein. This is not universal. There are some mutations that cause early frame shift or stop codon changes that are predicted to produce, not produce full-length protein. But for those that do simply cause one amino acid change, these changes have a destabilizing effect, impair the ability of the protein to maintain proper folding. And so the question is, can you identify molecules that would stabilize protein folding and allow them to function better? And certainly there are cellular proteins known as chaperones that have this function for all proteins as they're being synthesized. And there's some precedent in some disorders that small molecule, drug-like molecules, can function as pharmacologic chaperones. They're often small molecules that interact with the active site of an enzyme. So to do that, we've developed assays. And we wanted assays that were a little bit simpler and faster than the assays we developed using radiolabeled phospholipids. So we utilized some commercially available phospholipid-like molecules, phospholipid analogs, and experimented with whether we could use these substrates as measures of PLA2G6 protein function. And the bottom line is we can. These substrates work by having a fluorescent tag on one of the acyl chains connected to another quenching molecule. And when the acyl chain is hydrolyzed, you separate the fluorescent tag from the quenching molecule and get fluorescence. And in fact, we can use two different substrates, and the substrates show both A1 and A2 phospholipase activity of PLA2G6, and they're equally effective. So if we take that assay and look at purified recombinant protein, so kind of in a test tube, activity of an enzyme in a test tube, we see results with the fluorescent substrates that look very similar to what we've measured with radiolabeled phospholipid substrates. The wild type enzyme is quite active, but the enzymes containing INAD-associated mutations in some cases are quite inactive and have no measurable activity, and in some cases have partial activity, like the A341T mutation shown here. So we can use, so we can do real-time measurements. We can mix together enzyme preparations and phospholipid substrate, the phospholipid substrates, measure activity. And one of the things we first looked at is, in terms of a pharmacologic chaperone approach, is does ATP have an effect? In previous studies by at Dennis's lab had suggested this. And so we found the same, that we found not only for wild type protein, that if you add in ATP, you record a faster rate of activity, but also if you start with one of the mutations, R741W, and add in ATP, you see enhanced activity, not restored to the level of wild type, but in fact, we actually saw a kind of a greater effect in terms of percent increase on the mutant than we did in the wild type protein, which supports the idea. There is a known nucleotide binding site within the PLA2G6 sequence, and this type of result is kind of in some ways a proof of concept that there are small molecules, although ATP is not a drug-like molecule, there are small molecules that can improve function of not only the wild type enzyme, but also mutant enzymes. But we wanted to go a little bit further in terms of our ability to screen for other more drug-like molecules that could have a similar effect. And that includes studies not only with purified protein in a test tube, but also in live cells. And so we also used the same kind of approach to develop an assay where we can, in fact, measure the activity of the enzyme in live cells. It's complicated in part by the fact that there's a lot of other phospholipases in live cells, and so we have a fair amount of background activity. But in fact, we can see a measurable increase when we transfect and overexpress the PLA2G6 protein. And if we also kind of correlate that with ELISAs to measure just how much protein we are producing, to kind of normalize activity according to the level of protein production, which is variable with transient transfection and may be affected by the mutations as well. We can get live cell measurements that look a lot like, in this case, a lot like what we see with purified protein, suggesting that this that kind of measuring in vivo or in cells at least can reflect what we see in vitro, and that's encouraging for both assays. We also looked at an approach that would use cell lysates, thinking that this might be kind of a compromise in some way, that it might be a more stable preparation for screening, but might have all the cellular constituents that might help us understand or help us look for kind of other indirect effects of drug-like molecules on improving PLA2G6 activity. And so we did that measurement, and it looked same when we just took kind of sonicated lysates, with the exception of one enzyme, this R538H mutation. And so what you see here is, if you look at the purified protein or look at live cells, it's essentially inactive, but in these cell lysates, it was active. This was reproducible. And our best interpretation is that upon lysing the cells, you release, in this case, a cellular component that may have chaperone activity, which would further support the approach. However, we did not really see this for any other of the mutations so far. Another observation that kind of supports the approach that there are ways to stabilize protein folding for these mutant proteins is that we saw a reduction in temperature, which reduces the stress on protein folding, also had an effect of enhancing the activity of this one mutant protein shown here, the R741W. So if we look at it at 37 degrees, it has very little activity compared to wild type, but at 30 degrees relative to wild type, it has more activity in the different types of assays that we can run. Not as impressive for one other mutation, the A341T that I've been showing before, a small effect, but not quite as dramatic as what we saw for the R741W. So, one goal and one kind of initial target or goal in the project was to kind of use these assays, develop them to the point where we could do screening, and so with much effort to kind of optimize the assay, working with the high throughput screening center at Washington University, we scaled up the assay and adapted it for 384 well plates, and pursued a pilot, kind of feasibility screen using this assay. Could we identify compounds in a library, a collection of compounds? Could we identify a compound or compounds that might have the desired effect, would enhance the activity or increase the activity of the mutant protein? And we chose to, for our screen, a collection known as a spectrum collection, which contains already approved US and international drugs, as well as some natural products and other diverse compounds that had already been characterized to some extent, so that if we did have a hit, the work ahead in terms of optimizing such a hit for kind of use, clinical use, would not be as great. And so we completed screens with 2 different mutant proteins, which I had included in the data I showed you before. And so far in those screens, we did not succeed in identifying, the bottom line is we did not succeed in identifying a confirmed hit, meaning one of the compounds that would produce a significant increase in phospholipase activity. Are there compounds that produce perhaps very marginal increases? Possibly. We'd have to do more experiments to really characterize this further. But so far we have not seen kind of consistent reproducible effects of a given compound. And I will note that one screen, the R741W, did perform suboptimally in terms of the screening assay. And so although we did not see any hits, we, if we had the time, would like to pursue that further as kind of understanding feasibility and proof of concept for this approach. In terms of future directions, we are thinking about further assay development, including characterization of additional mutations. We want to understand how broadly applicable this is. Obviously, it's not going to work for mutations that don't produce a full-length protein. And so that does mean it essentially could be applicable to only a subset of INAD patients. But the question is it really even specific for only a subset of the missense amino acid change mutations? And I think we need more data to understand that. There are additional ways to screen, including screening with live cell assays. We like screening already approved drugs, potentially capitalizing on the ATP effect. We could screen, there are analogs that have been developed for other targets such as kinases that we could screen as well. And ultimately we'd like to, if we do get a hit, we'd like to look in neuronal cultures and see whether it also affects this mislocalization effect in primary neuronal cultures that we see. Could this have other applications in terms of direct, a direct therapeutic effect? And I think it could. And so I think it's worth thinking about this in the long run as we think about gene therapy and potentially enzyme replacement therapy, is that if you have suboptimal production of the native protein, either through gene therapy or if you're trying to deliver recombinant protein as an enzyme replacement therapy, such approaches to kind of help stabilize and improve the function of the PLA2G6 could enhance these approaches. And so, even producing recombinant protein in vitro can be helped with this approach. And then finally, I'd like to just acknowledge the people in my lab who have worked on this project. And most of the work was done by Chandana Badala, who was a postdoc in my lab, with also help from Joanna Luo, who worked in my lab as an undergraduate student. And I want to thank our supporters for this project. And for this particular project, it was primarily the Jasper Valentine Foundation, Ainsley's Angels of America, and the Phillips Herschelman, Lemaire, and Thompson families. And with that, I'll take any questions.
Thank you, Paul, for an excellent talk and a very nice kind of broad overview of how to approach the PLA2G6 conundrum. We have time for a couple of quick questions. Is there a crystal structure of PLA2G6 that could guide compound development?
So there is a crystal structure of one of the two isoforms produced, and I do think, yes, that's actually a good thought, and that is the kind of thing that could guide compound development, particularly if we were able to get a hit, but potentially capitalizing on that if you can characterize binding sites for molecules like ATP. You know in that case, if you have a binding site and how a particular ligand or drug might fight into that binding site, that can guide you in terms of optimizing structure of a drug-like molecule. So it's a good question.
And do you think, question from Sujong, are there other PLA2G6s that may be able to compensate for G6?
Yeah, I've thought a lot about that actually. And there's precedence in other disorders for looking at those approaches, including Susie's approaches for PCAN. And I don't think it's quite as simple, unfortunately. To my reading, it's not quite as simple. There's a family called PMPLA, or patagen-like phospholipases, that contain, I believe, 8 different genes and enzymes. The closest one is known as IPLA2 gamma, which is roughly about 50% related at the amino acid sequence. We've actually looked at that gene with a collaborator and studied knockout mice for that gene, and it is different in that it's localized almost exclusively to the mitochondria, and the deficits we see in the knockout mice are primarily in the mitochondria, and with its effects on cardiolipin metabolism. So could it partially compensate? Because there's some evidence that PLA2G6 also functions or has some role with mitochondrial function. Yes, but I think it would only be kind of partial overlap in this case.
And just one more question, because I think this is a very pertinent question, Arcangelo. So is asking, have you tried to correct PLA2G6 deficiency by expressing C19-OFF12 in mutant cells?
No, I haven't. It's a good question.
Yeah. Okay, thank you very much, Paul, for an excellent presentation. We'll move on